All right. I'm going to introduce carbohydrates, and then what I want you to do today is start making a molecule, one saccharide, and we're going to get a certain way into this, and on Monday we'll come back to that molecule you make today. So you're going to work in groups of, it'll take me about 35 minutes to get to that. And then you'll make this molecule, and when we come back on Monday, we'll do some more things with that molecule. You're going to see that these saccharides can cyclize. So we're not going to convert it into its ring structure today, but hopefully we'll get the molecule made so that we can finish it off on Monday. Okay? All right. We are done with organic. Okay, no applause, no applause, no applause. Applaud yourselves. Applaud yourselves. You've gotten through 6 chapters of organic chemistry, and now we're going to go through, I don't know what it is, 5 or 6 chapters of biochemistry. That's what this course is made up of. Half organic. It's an introduction to organic chemistry. It's an introduction to biochemistry. And in the biochemistry section, we're going to concentrate on the 4 major classes of biochemical molecules: carbohydrates, lipids, protein, and then nucleic acids. All right, so we're going to, chapter 16 will be on carbohydrates, chapter 17 lipids, chapter 18 and 19 will deal with proteins, and then chapter 20, nucleic acids. Okay, so that's where we're headed. All right, carbohydrates are the building block, and usually that's what we will look at in each one of these things. We'll kind of break it down into, okay, what's the building block of carbohydrates? It's saccharides, monosaccharides, individual sugar molecules. That's the building block of carbohydrates. Okay, we don't need to read all of that, but this is, you can look at that slide and it's kind of a little checklist. This is what we want to get through in this chapter. Know the difference between mono, di, and poly. Naming monosaccharides according to functional group and number of carbons. Know what a chiral carbon is and why it is so important. Explain stereoisomers, identify monosaccharides as either D or L. Know how to draw Fisher projections. So we're going to need to learn how to draw this Fisher projection and Haworth projections. This relates to the saccharide when it's in the chain or open form of it, and the Haworth projection is when it cyclizes into a ring. How do you draw that? We're going to learn those things. Benedict's reagent. Common disaccharides, table sugar, maltose, lactose. So that's kind of a summary of what we're covering in this chapter. Carbohydrates. We use them. We use this generally for energy. You get about 4 kilocalories or 4 nutritional calories, capital C. A capital C, that's 1,000 little calories. 4 calories of energy per gram of sugar. I should change this slide because this number keeps changing all the time. You get into the a keto diet, and it's very low in carbohydrates. So that number is up for debate and discussion, and some people would say that'd be way too high. All right, here's the most famous monosaccharide, glucose. and we'll get familiar with what we're talking about here in a minute, but there's glucose. And if you think about it, let's think about this for a second, because this is what you're familiar with. Let's put a 6 carbon, so that's 3, 4, 5, 6 carbons. And if this were, those were all hydrogens, all right, you'd have a fairly nonpolar molecule, nonpolar molecule. And it would it be soluble in water? No. So this is hexane over here. I'm just putting it here to contrast for a minute. Hexane is nonpolar, not soluble in water. But then when we start talking about saccharides, there'll be one exception to this, but generally you have an OH group on all but one of the carbons, and then you have a carbonyl on the last carbon. It's not always at the top. But so you have a carbonyl and then OHs on everything. And what do OHs do to a molecule? This is nonpolar. What do OHs do to a molecule? Makes it polar. And so at every site along there, you have an OH. You have a polar bond, which is the reason why glucose is so soluble in water. And it flows, it transports very naturally and easily through the bloodstream, because the bloodstream is basically an aqueous environment, and glucose is very soluble in water, and this is why it's very soluble in water. Every single carbon has got an oxygen on it. Makes it a pretty polar molecule. And so everywhere along here, if you look at that bottom carbon, what do you have? You have an OH there. I've collapsed it together, but what that means is that it can hydrogen bond with water, these OHs. All up along there, the water feels real comfortable around it. It feels real comfortable in water because they are both polar. So there's one example, glucose, erythrose. You can see they can vary in chain length. They're going to have an OH on every carbon except one of them, one of the carbons. All right, so generally we look at a monosaccharide. You might see it drawn as kind of a ring like that, because what's happening here, let me just go back one slide for a second. What's happening in that ring is that this molecule is forming a ring with itself. We'll get to see that. We're going to concentrate on what I would say the open chain image or formula of these saccharides, and then we're going to cyclize them and see how that happens. But when they do cyclize, this is kind of a quick way we draw them. It's a 6-membered ring. There's actually an oxygen right in there. It's not all carbons, so that's not hexane. That's 5 carbons and an oxygen in a 6-membered ring. That's a monosaccharide. A disaccharide, 2 of those linked together, is a disaccharide. A glycosidic bond, we'll see what that bond is in more detail later. Polysaccharides, I'll come down here to polysaccharides, many saccharides, 20, 50, 100, 1,000. You got a polysaccharide, but in between the disaccharide and the polysaccharide, get familiar with this little prefix right here, oligo, an oligosaccharide. It's not a poly, it's not a di. 3 to 10 units. It might be 3 to 11 or 3 to 12, but it's not many. You know, it's not so many that you start talking about poly. All right, so an oligosaccharide. This will even show up in some papers about the sensitivity some people might have to oligosaccharides versus mono or poly. Oligosaccharides. A homopolysaccharide. let's say those are all glucose. Then you have a homopolysaccharide. If you had glucose and fructose or a mixture of different saccharides in your polysaccharide, you'd have a heteropolysaccharide. And then ordinarily this is going from carbon number 1, this is carbon number 1, this is carbon number 4. 1, 2, 3, 4. So this would be a 1, 4, 1, 4, 1, 4 linear polysaccharide. So here's a 1, 4, but then also you have a 2 something or other, 2, 5 linkage here as a branch. So this one right here is branching off to another branch of the polysaccharide. So just be kind of familiar with that sort of thing. All right. This is an important slide. Yes?
One quick question on the monosaccharide. I missed where you said that it wasn't all carbon. One of them was
an oxygen.
An oxygen, right?
Yeah, and we'll, I'm just kind of introducing. We'll really look to see where that oxygen is and why that oxygen is there and how it cyclizes. You're going to want to know that. I would star this slide just because it really does kind of summarize how to name different ways of naming saccharides. Okay, now what they're concentrating on here are two things. What is the chain length? How many carbons are in this monosaccharide? And number 2, where is the carbonyl? All right, now there's really only two options that we generally see for that carbonyl. I don't know any exceptions to this. There might be, but generally the carbonyl is either going to be at the top, and that will be carbon number 1. And what functionality is that? When it's on the end, it is a aldehyde. Or it's on carbon number 2. It's not on the end, it's down one. It's not just anywhere in here. It's on carbon number 2, which is what kind of functionality? Ketone. All right, you see how you drag your organic chemistry right into biochemistry? Don't forget your organic. Biochemical molecules are organic molecules. They're naturally occurring organic molecules. They are biologically active organic molecules, okay? So everything you've learned in the organic section is going to be helpful here. All right, so those are aldehydes. So we would call that an aldose. The OSE ending is pointing you to the fact that this is a carbohydrate. And it doesn't necessarily mean it's a mono. Sucrose is a disaccharide. Okay, so the OSE ending is telling you this is a carbohydrate. If it's got the aldehyde functional group on it, it's an aldose. That's not a very specific name. It just says it's an aldose. The carbonyl is on the top, or it's on the end, and we draw them like that. We draw the carbonyl towards the top and draw these generally vertically downward. How many carbons are in it? 3. It's a triose. So that is an aldose. That is a triose. It's an aldotriose, putting that all together. And its name is, none of those are its name. None of those are its name. All of those are just kind of, what group does it fit in? But its name is glyceraldehyde. That's the name of that monosaccharide. That's an important one.
Where did they get glycer from?
That's a good question. You already seen this molecule. Has 3 OHs on it. This is glycerol. That's glycerol. That's where they're getting glycer from. From this is glycerol, and if we just put that at the top, you have glyceraldehyde. Okay? So now that begs the question, why do they call that glycerol?
Yes.
And now you're starting to act like a 2-year-old. You're asking a lot of why questions. And why? And those are good. Those are very good. But sometimes you just have to say, I don't know. Why do things fall to the ground? Okay. No, all right? Is that good enough for right now? Okay. Glyceraldehyde. This is one you'll learn eventually. But let's concentrate on this naming system. So now here's glucose. It's an aldose, just like glyceraldehyde is an aldose. It's a hexose. It's not a triose. It's a hexose. 1, 2, 3, 4, 5, 6 carbons. It's an aldohexose, and it has this name, glucose. Glucose. Don't worry about the D's right now, but it is D-glucose. But we'll figure out where that comes from in a minute. Okay, now here's glucose. Here is fructose. 1, 2, 3, 4, 5, 6. So it's a hexose, but it's not an aldose. It's a ketose. So it's a ketohexose. And it is fructose. Yeah, where does that name come from? Fruit. Okay? It's in grapes. It comes from fruit. Fructose. That's glucose. Let me see what we're going next here. Time to pray.
Yes.
This is exam 3. Bring hands. I forgot my mirror. Does anybody have a mirror? I can't. Oh well. I didn't know I was going to get into this today. I usually bring a mirror and a couple molecules.
We can do it next time.
No, we kind of need it. We kind of need it. So, that's all right. We can do this real quickly. Whitney's going to help me. Whitney,
How big of a mirror do you need?
Whitney, put all of these on there, and then put I don't have another yellow one. Amy's going to help me too.
Okay.
Put all those on there. Here you go, Amy. Catch. Good catch.
Color order?
No, just put them on there. Okay, here, put these on there. Okay. Let's get to the praying hands for a second here. Your hands, well, let's get these molecules and then we'll talk. Quickly, I need that molecule. Fast. Real fast.
Oh, you're making me nervous.
All right, I like it. Oh, let's see. Wait, what do I got here? I got a white ta-ta-ta-ta. So I need to change that one. The white one.
Sabotage me on purpose. What were you doing here?
All right. Okay. Let me just look one more time here. All right. Whitney and Amy made these two molecules. It's got a carbon. Tristan?
Yes.
These two molecules here. I want everybody to tell me, think about it, are they identical?
Yes. Yes.
No, they're not.
Is it trick? Yes, they are.
They're identical?
Yes.
Okay, we got a carbon, and you would say, what are these things on the carbon? Well, just think of them as 4 different things. Any 4 things. It could be a hydrogen, an oxygen, a nitrogen, and something else, a chlorine, whatever you want it to be. But they're all different. But this yellow is the same as that yellow. This white, if these are whites or hydrogens, which they usually are, those are hydrogens, nitrogens, oxygens. So the reds are identical, the yellows are identical, the blues are identical, the whites are identical. The carbon is carbon. So we got a carbon with 4 things on it. We got a carbon here with 4 of the exact same things on it. Are they the same? Yes or no?
Yes.
Yes?
They're
No?
the same.
What would be the acid test? I shouldn't use that word in chemistry, but what would be the fundamental test we could do to say whether these are identical and do not need separate names? What could we do? We could do it right here in class, right now. What could we do right now to tell whether these are absolutely identical? No? Meaning? No, these will have exactly the same molecular weight. They're made out of the same stuff. Remember what I've said when we would draw different molecules, and I said, well, I don't care whether you draw it this way or that way, and those are superimposable on one another. Do you remember me saying that? If 2 things are superimposable, they are identical. Well, let's see if these are superimposable. What does that mean? I'm going to put the carbon on the carbon. I'm going to put the blue one on the blue one, the white one on the white one. Is the red one on the red one?
No.
No. It's on the yellow one. And the yellow, okay, well let me flip it around and get the yellow one on the yellow one and the red one on the red one, and then the blue ones and the white ones aren't on top of each other.
So they're not superimposable.
They are not superimposable. So what does that mean? They're different. Geometrically, the geometry around this carbon is different. These are 2 different things. Now why is that up there? Why is that up there?
Because your hands are the same thing. They have the same amount, but they're similar.
Your hands are not identical. They are complementary. Your left hand and your right hand complement one another. And if this were a mirror, if this was a mirror, and I put my right hand on the mirror, it would look just like my left hand. It would look like my left hand. That's why I wanted to bring a mirror today, to just show you that your right hand is a mirror image of your left hand. And these molecules are mirror images. If we had a mirror right here, let's see, how do I do this? How do I do this? Put that up there. Yeah, like that. Okay? Can you see if I had a mirror right here, that these 2 molecules would be mirror images of one another? Okay? They are mirror images, but they are different isomers, and these are stereoisomers of each other. Stereoisomers. Just like your hands are mirror images of one another, those molecules are mirror images of one another, and this ends up being very subtle difference. There's a very subtle difference between these 2 molecules, but a very important difference biochemically. Because what will happen with saccharides, what's going to happen with amino acids, and other molecules, biologically active molecules in your body will use one of the isomers, and nature will produce one of the isomers and not the other one. It will exclusively produce one isomer, and I wish you could appreciate how amazing that is. I mean, I'm amazed by that as a chemist. I can't go across the hall here and make D-glucose. But the trees are out there making it all the time. Nothing but D-glucose. All right? Now, can I do it in the lab? Yes, I can do it in the lab, but I need the help of bacteria. I need the help of something living to make D-glucose. Because biologically active living systems that can make proteins and can metabolize things in a very sophisticated manner can make D-glucose. But I can't just go there in a beaker and mix up stuff. I can make glucose. I can make glucose over in the lab, but it's going to be a mixture of the two isomers. All right? The trees are out there, the dumb old oak tree out there is making D-glucose all the time. Shows you how smart I am. I can't go across the lab and do what that oak tree has been doing all the time. All right, so I just want you to stop and, well, you'll see more of this. So mirror images. We're talking about mirror images, and I do want to tell you about glyceraldehyde. Glyceraldehyde is actually going to be our reference molecule. We're going to get familiar with what D-glyceraldehyde looks like, what L-glyceraldehyde looks like, and those are going to be our references for figuring out any monosaccharide. We're going to compare it to glyceraldehyde, and we're going to be able to tell whether it is a D or an L isomer. So this is our bottom line reference molecule for D and L. And the biologically active one is D. And you're going to find that with all these saccharides. It is D-glucose, D-fructose, D-glyceraldehyde that is biologically active. And D-glucose. Yeah, there's some kind of really wild exceptions to that, like something that's toxic or something that some sort of bacteria might make an L something every so often, but 99% of the time
It's all saccharides, it's just monosaccharides.
Well, the rest of them, the di and the poly, are made up of the mono. So if you start out with D-glucose, you're going to end up with glycogen. Glycogen is the polysaccharide of glucose, and it's going to be made up of all D-glucose. I'll tell you what, let's we've introduced this. When I come back, or when we come back on Monday, we'll have the D and the L-glyceraldehyde. But what I want you to do is to get into like groups of probably 3 or 4, because I probably only need about 5 groups here. I'm going to give you a set of a modeling kit. I want you to make glucose. This is what you're going to make. Yeah, and I actually do want it to be D-glucose. Well, you make glucose, and we will make sure it's D at the start of our class on Monday. So you want to make 6 carbons long. These are all carbons. And you want to put it, these are going to be oxygens, carbons. Oh, folks, I want you to use these bonds for everything except that bond up there. Have we done this before? So that bond up there, you've done it in lab anyway. That bond up there, use these bonds here. So get into your groups and work as a team. If you run out of something, there'll be an extra box up front here. You guys want to do this together?
Why do you have to text it to me?
Is this your team here? Okay, what
We have to do that.
Maybe I can talk to you later about this.
Well, you don't have any reds? Okay, come up and get some.
Should I just take them over here or what?
I screwed up. I need to go over and get my associates thing taken care of because they're here. Today's the last day, and I got to leave after my next class.
Okay.
So I'm gonna get with Patty.
You need about 6, yeah.
I'm gonna get with Patty afterwards and figure out what I missed.
All right.
All right, thank you.
Oh, yeah, that's good.
Okay.
You want chapter 16 homework?
Oh yes. So about hospital things create like all animals except for us and getting things do vital things. That's amazing.
Yeah.
Like more scientists.
Yeah, well,
You can't avoid it.
yeah, I do say a little bit about it when we finally get talking a little bit about the original life, the evolution of life.
Did you see my thing?
I mean, when you get the DNA, I mean, this is child's play.
Right.
Compared to DNA.
Oh my gosh. When I first
But I mean, but this is almost impossible for us. And then you get up to DNA and you say, whoa, the level of complexity here is just overwhelming.
and everything that your body has to do insane to do it. They think it's just it boggles me.
Yeah, well, I have a firm belief that this is the best chemist. There's no chemist like him. And I'm amazed. That's why I say stop and be amazed at what you see. You hear this, you hear all kinds of things in your career time. You think, oh, well, that's nothing you learn. Oh, wait a minute, stop a minute. This is incredible. This is a very subtle difference, and your body can tell the difference.
I mean, you can argue all day, I guess, about which, but technology is
Exactly. Very good.
Okay, so I need a.
Can we get 2 more of these?